I just want to make sure everyone knows what I'm talking about today. I'm going to be talking about how clinical trials are done, okay? Why they are done that way. I'm not going to be talking about specific studies. There's actually too many studies to cover, and we don't actually have updates until companies actually release them, because they're all run by companies. So if you're expecting to hear about specific clinical trials, I will not be talking about those, okay? But if you want to understand a little bit about how clinical trials are done, why we do them, the specific methods, I'm going to be covering that. And I think this is an important topic for people who are considering participating in clinical trials. Then you can kind of understand where we're coming from. I am somebody who sees a lot of patients with muscle disease, and I also participate in a lot of clinical trials. If you go into a clinical trial understanding what I'm talking about today, then it'll help the investigator, help the team, help the company, and ultimately help your disease to really help find effective treatments. Okay, so that's what I'm going to be talking about today. So before I begin talking about how clinical trials are done, I'm going to just really quickly talk about two different kinds of clinical trials, two different kinds of drugs that we investigate in clinical trials. Okay, just very simplistically, we can talk about new drugs, and then we can talk about repurposed drugs. Okay, so a new drug is something that's never really been given, at least on a commercial level, to patients before. So it's been given only on an experimental basis, okay, or maybe never given to humans at all before. So sometimes we start with new drugs, and we're in the process of establishing everything about that drug. Okay, so we have to start off with safety. We have to establish what the safety is. We have to make sure that the drug is safe at the levels that we want to give it to our patients. Okay, we don't want to be, well, let's say it works really well in a petri dish. Okay, well, the next thing we do is we give it to animals, and then we give it at reasonably high doses, and even doses that are higher than what we would need to establish to see how much buffer we have. So let's just say that the drug has to be given at 10, just making up a number, in order for it to work, we think. We think it would work at a level of 10. Well, a lot of times they'll do experiments in animals that take it up to 100, or even higher than that. They want to see, how high you can go before you start having toxicity, in the animal. All right, and figure out that range of safety before we start giving it to humans and to people. Okay. And then what comes a little bit later is, well, we think it works in a petri dish because there's some muscle cell or some lung tissue that you have in a petri dish, and then you can do some experiments that seem to suggest that something's working. The immune cells are backing off or something like that, whatever it is that we're testing. Then we start testing them in animals, and then if it works in animals, if we have an animal model. Unfortunately for myositis, we don't have a whole lot of animal models, not real animal models, but we try to make some animal models. Animal model is basically an animal who has the disease that we're trying to treat. Well, that's difficult, and usually we use mice. Once we can test them, at least safety and efficacy in animals, then we start moving to humans, and then we try to establish efficacy. Does it work? That's the question. Okay. And this is generally disease specific. And that kind of depends on the investigators and perhaps the company that's sponsoring this clinical trial. They define the disease. So they can't just say anybody who has autoimmune disease. They'll usually define something that's considered a disease that we can talk about, like dermatomyositis. Should we get really specific and say which antibody? That's probably up to debate right now. Okay, but sort of the way we want to define what disease these patients have, and then they would be the ones included in these clinical trials. Then there's repurposed drugs. So these are drugs that already are on the market for some other reason, for some other disease. Okay? And the data that we have, actually, usually we have a whole lot of information on their safety because we've been giving it to lots of patients. So we have established safety record, even historically when it was first given to animals, and then of course all the clinical experience that we have in humans that have been treated with this drug. And perhaps you want to say, well, let's now see if this drug would work in a new disease. And that would be a new disease. Generally speaking, these drugs are sponsored by companies. These drugs, when they're being restudied, may or may not be sponsored by companies. Okay. These companies are trying to establish something totally novel in this left column. Here, a company may have something to gain in terms of being able to market that drug if they want the FDA to give an endorsement and say, yes, this has been proven. We approve this drug for use in this disease, in addition to that other disease that we already approved it for. Okay. Sometimes companies will do that. Sometimes companies are not interested in getting that endorsement, but if there's a bunch of doctors that publish a paper and says this drug works in this disease, then people will start using it. So, just to give you some examples, new drugs in myositis, abatacept, arimoclomol, these are things that we sort of briefly mentioned today. A repurposed drug would be like rituximab. So rituximab was originally designed for certain kinds of cancers, right? Lymphoma and so forth. But now we're finding that if we go after B cells, that can actually be used for a number of autoimmune diseases. So it's really had a number of different studies that have been used as a repurposed drug. So let me spend a little bit of time talking about what the FDA is looking for, which is probably a higher standard in some cases. But what we're talking about is something that is, from the very beginning. So all the way before it's been given to humans, before it's given to animals, it's in this discovery phase where we're doing things in petri dishes, where we have something that's growing that may indicate something about your disease, and we start testing them. And sometimes we can screen through these, what we call them libraries of molecules, of compounds, and they can just put them into a lot of different petri dishes, mini petri dishes, tiny ones, okay? And then you can screen them to see if one of these drugs might give you the intended effect. Be able to, calm those B cells down, calm those T cells down, or whatever it is, affect autoimmune disease. Then you can move over into more preclinical studies, which is usually in animals. And then what companies do is they submit what's, called an IND, investigational new drug, which gives the company and anybody who works with the company, including doctors that are contracted to work with the company, the permission to start giving it to humans. So they have to pass that bar, say, we think that there's enough information here that says it's reasonable to go ahead and start doing experiments in people, okay? And I like to use that word experiments because it is an experiment, okay? When we're doing clinical trials, I want you to realize it's an experiment, you may be getting the drug, you may not be getting the drug, it may help you, it may not help you, okay? And then it goes through these phases, and I'm going to talk a little bit more about these phases a little bit later. But phase 1 is usually small. Sometimes it's given to healthy people. Sometimes it's given to people who have diseases. But it's the first time we're giving this drug into people. Then we go to phase 2, which says, hey, we're confident that it's not killing people, it's not doing anything seriously bad to people, so then maybe we can give it to more people, make sure it is safe, and then start collecting a little bit of information about whether it works. And if that looks good, then we go on to a really big study called a phase 3 study, which is really designed to show that this drug works. And of course, continue to collect safety information. If everything looks great, then the company may submit a new drug application. That's what an NDA is. And then the drug is approved. And many companies would be required at some level to continue to collect safety information. They're all supposed to collect some safety information. It's just how rigorous they have to be about collecting that safety information. This is really a long process. I mean, when we talk about how come there aren't any new drugs, I mean, this is typically 10 to 15 years. 10 is pretty optimistic. 15 years, is a little bit more typical. You can see that on average, we're talking about trying out 10,000 molecules before you get one drug that's going to get approved. Okay, 10,000 molecules screened will eventually lead to about 5 drugs that we give to humans, and usually about only one of those 5 is going to make it pass every test. So this is a weeding out process. But of course, we only want drugs that work. We don't want drugs that don't work. We don't want drugs that are not safe. And so this is an important process. So I talked about the investigational new drug application, and then there's also a new drug application, which is for the final approval. And so these are when scientists work over here on the left. Clinical trialists, I'm a bit of a clinical trialist, work here in the middle. And of course, this is when doctors are in regular practice. Okay. And more and more, the pharmaceutical companies get involved. Okay. Sometimes over here, there are certain kinds of pharmaceutical companies that are called biotech companies, and they're smaller. They're really driven to discover new things. So that's where you hear about those types of companies. So what does the FDA want? Well, we have a pretty significant experience of drugs in the past that perhaps moved through the approval process a little too quickly. You probably remember thalidomide. There's this drug that was given back in the 60s, and a lot of babies were born with a lot of defects. And it was a wonder drug, and it's still a drug that can be used. It's just we now know a lot of the safety limitations of thalidomide. And so that really made the FDA a lot more careful about making sure we know everything we reasonably can about a drug before it's allowed to be taken to market. So is it safe? Is it safe in animals? Is it safe in humans? And then does it work? Is it effective? Usually we want a placebo study, meaning that patients are receiving either they might be receiving the real drug, or they might be receiving a simulated, version of the drug, a sugar pill or something like that, as people figuratively call it. And then we have to establish that the drug works, and it works in a clinically meaningful way, meaning that it matters to the patient what it is that we've made them better about. Okay. So I'll talk a little bit about clinically meaningful. I mean, if I said that I don't really make my patients better at doing their daily living activities, but they could stand 10 seconds longer than if they weren't on the drug. One would argue that maybe that 10 seconds, that's not enough of an important effect on the patient, So just to make up some effect. Maybe there is some real effect, but that effect is not clinically meaningful. And we also have to make sure that there are enough patients that really prove beyond a reasonable doubt that this drug is working, not that it's some random thing that happened, okay, the way the study was done. So placebo, it's a non-active drug. So in injections, sometimes we inject saline, There could be many different ways to make a placebo. And it's given to simulate the active treatment. So meaning that the patient will not know whether he or she is receiving the real drug or the simulated drug. And a lot of times we do this in a way where even the doctor who's running the study the investigator doesn't know what you're receiving. So if you were participating in a clinical trial at my institution and I was in charge, most of the time I don't even know what you're getting. Okay? And that's what we call double blind. Double blind means that neither the patient nor the investigator who's doing the evaluation, who's doing all the tests on the patients, okay, knows who is getting placebo or who's getting drug. And there are a number of different ways of designing it. Sometimes you can just say half of the patients will get drug, half the patients will get placebo. Sometimes it's not okay to give placebo, so you'll give what's a sham procedure. So sometimes when we do injections into patients' spines, we don't want to put the needle all the way in. We just pretend to put the needle in. We put it in the back and don't go all the way in. That's called a sham. So that's kind of like a placebo. Sometimes we do what's called a crossover design. So you'll hear like these types of things where, let's say patient here gets the drug, these patients here get the placebo. And then just to make things interesting, you kind of swap them halfway through the study. Okay, and you would be able to see that perhaps if drug A is working and drug B is the placebo, that these patients get better and then perhaps they get a little bit worse here. Or likewise the other way around, they don't get better here and they get better after they get on drug A. But if that pattern holds up in almost everyone, then you know that this effect is truly related to the drug. Because the placebo effect can be very real. If you say that somebody thinks they're getting the drug and then you just say, okay, can you walk down the hall? Let's say that's my test. Can you walk down the hall? And you think, oh gosh, I'm getting that drug, I'm gonna get better, I'm gonna walk down that hall. Whereas before you would struggle, but then you try a little harder because you're more motivated and perhaps you seem to perform a little bit better, And if everyone does that, and we don't have a placebo, then it's possible that could create a false effect. Okay. There's another way of doing it. Everyone's on the drug, and then half of them stop the drug, the other half stay on the drug, and you compare these two groups. So we're always comparing these two groups. But what is the test? How do we decide if a drug's working? We have to come up with a test. We have to decide, should it be like walking down the hall? Should we have you walk down the hall back and forth for 6 minutes? What they call the 6-minute walk test. Should we have you do push-ups? Whatever it is. You have to decide. That's what we call an outcome measure. Okay. So outcome measures, there are different kinds of outcome measures. There are some where we, as investigators or as providers, as health professionals, perform the test. And there are other ones where we ask you a whole bunch of questions. And perhaps we just give you a survey and you fill out on a scale of 1 to 10. How do you feel about this? How do you feel about that? Okay. And ask perhaps a bunch of questions, 5, 10, 20, 30 questions. Those are what we call patient-reported outcome measures. Now this has really become important to the FDA because the FDA wants to know that patients actually are feeling better. If this drug is working, and that the patients are actually saying, their lives are better, or that they're better than the people who didn't get the drug. Okay, so it is subjective, but it's subjective in the most important way, in the best way, because it's subjective about to the patient who's actually receiving this drug or placebo, to assess how you feel. You as the patient, if you're participating in a clinical trial, how you feel about being on this drug. And so that's thought to be pretty clinically meaningful. If you see a real consistent effect of people who are treated versus people who are placebo, and they tell you these are things that are important to me, and I do feel better about it, then that's clinically meaningful. There's also provider assessment, so I can just say, hey, I want you to do this task. I want you to do push-ups. I want you to do jumping jacks. I'm just making stuff up, okay? But any one of these could, in theory, be a test for something like neuromuscular disease. Okay. It's often designed by researchers. It's objective, but it has to go through a whole set of rigorous studies to make sure it's valid. Like this is something that's actually useful in this disease. So I'll talk about that in a second. And the other type of outcome measure is a biomarker. Okay? So maybe certain kinds of blood tests that might tell us if a drug is working or not. Usually biomarkers are not good enough in and of themselves. You can't say, oh, but the biomarker is working. Okay, well that usually does not get a drug approved. But it is nice to see biomarkers move the right direction. But usually one of these two, and historically it's usually been one of these types of tests. But more and more this is playing a more important role in getting drugs approved. But when scientists, when the FDA, when everyone's looking at these studies and they're looking at all the data, usually we want to see most of these things lining up and going the correct direction. You don't want to just define some biomarkers that don't mean anything, that have nothing to do with the disease. You guys have probably seen this cartoon before. Looking for the keys right under the lamp. Well, where'd you drop the keys? Over by my car. Why are you looking over here? Because the light's better. Okay, here. Okay. So we don't want to do clinical trials over here because it's easier. We want to do clinical trials over here because that's where the money is, right? Because that's actually where you're going to see it. So we want to do tests that actually capture the disease. We don't want to do tests that do not have much to do with the disease. So outcome measures, they need to be validated through studies. Typically we have to do studies, natural history studies, meaning we get a bunch of people who have this disease. These people who are in the studies are supposed to represent everybody who has this disease. So you don't want to just get the best ones or the worst patients. We want to get people across the whole spectrum, okay? And see how these outcome measures change, as they live with this disease. And in particular, this would be important for something like inclusion body myositis, where currently we don't have particularly powerful drugs that can actually change the course of the disease. And so we have to understand how inclusion body myositis is changing as patients live with the disease. So the FDA has determined that they only want outcome measures that are clinically meaningful. That means that the measure must be something that reflects life's activities or quality of life. And the degree of improvement has to be significant. So what's clinically meaningful? Well, here's a list of things that you might think about. Survival. Yeah, if you're talking about a disease which is possibly, going to terminate somebody's life, then survival would definitely be what most people would accept as clinically meaningful. Being able to breathe, that's pretty clinically meaningful. Swallowing, clinically meaningful. Movement, probably, if you talk about movement that's important movement, you don't want to focus on movements that people don't care about, like being able to move your ears, but let's say moving your eyes or being able to walk around, those are clinically meaningful movements. Fine motor skills, probably, being able to use your hands, being able to write, being able to type, those types of things. But, for instance, muscle mass. You might say, well, my muscles are shrinking because I have inclusion body myositis, for example. And, it would be great if you had a drug that would just make them grow back. But let's just say they grow back, but you don't get any stronger. Or you aren't able to use the muscles as well. You don't notice that you're any stronger, okay? Then that may not be clinically meaningful. So muscle mass in and of itself is not necessarily clinically meaningful. And then blood tests, biomarkers, for instance, that may be supportive, but that may not be enough in and of itself to convince the FDA that this is a drug that should be approved. Okay? So usually when we do clinical trials, there's what we call a primary outcome measure. Like you're going to put all your money on this one particular thing. I'm going to put all my money on swallowing. Or I'm going to put all my money on breathing. Okay, I'm going to tell the FDA, what we think is that this drug's going to make people breathe better. And also it'll probably make the blood tests look better, it'll probably make their movement better, but the one thing that we're really, really going to put our hand down is that this is going to help people breathe. Okay, then you do the clinical trial and that breathing measure better come out right. Okay, and then most of the other ones hopefully will also light up as well. So natural history is when we follow patients who have the disease and see what happens as they live with the disease. And it could be with treatment or without treatment, depending on how it's defined, but we usually capture these outcome measures. And sometimes if we have good natural history, meaning patients who are not on any drug that's not on any of the drug we're about to give to patients, that could sometimes be substituted for the placebo arm. So this is an example of an ideal outcome measure, sort of, Let's say through time, somebody's strength with treatment is good, and then the strength declines if the patient doesn't receive treatment. Or the opposite, let's just say without treatment, it's flat, and then with treatment, it goes up. Okay, you want a separation. Whatever this is, how fast you can walk, if you can do push-ups, how many push-ups you can do, that kind of stuff. Whatever it is the outcome measure and the degree to which you improve or maintain your strength. This would not be an ideal outcome measure. So you could even have a real effect. But for instance, you're testing a muscle which, I'm focusing on muscle because I'm a neurologist and I look at muscle, but you can always talk about other types of outcome measures. But we're looking at a muscle that doesn't change a whole lot, okay, with or without treatment. And the drug might work, but we're looking in the wrong place. There are keys. If you look in the right place, you'll find keys, okay? But stop looking under the lamp, right? Just because it's easier, right? We have to look where we think we're going to find the keys. So we have to look where we think we're going to show an effect that's clinically meaningful, that has something to do with the disease. We have to make sure we recruit the right patients. Inclusion, exclusion criteria. This is so the study can run smoothly. You can complete the study relatively quickly. Nobody wants to do clinical trials for 10, 20 years because, if we have to wait 10, 20 years for this drug to show an effect, that'll be a long time for all of us to be waiting to be able to use these drugs. So everybody's impatient. I would say the patients are impatient, the doctors are impatient, the companies are impatient, okay? Everybody wants a drug approved in a few years, okay? And a lot of times, you need a couple of years to go through these studies. You don't want to start a phase 1 and then 20 years later you're still doing phase 2. So patients are recruited and selected because they're going to likely establish and show the outcome measures that will show a difference between treatment and placebo. So just to give you an example, if I recruited, I keep on picking IBM because it's an easy target, okay? But if I took an IBM patient who has very, very mild weakness, and then I enrolled them in a study and then they got worse, or the patients who got placebo got worse, but the patients who got treatment stayed the same or got better, you would see a nice separation. Okay. But if I took IBM patients who were more advanced, and the progression was already kind of down here. So I recruited these types of IBM patients. Okay, let's say the drug works. There is a separation, but it's not a big separation. And if it's not a big separation, then you'd have to recruit a whole lot of patients to be able to show that works. Okay. Because then you need statistics. Yeah, question?
Is it usually different groups that do, like you'll often hear when people are asking questions of like the panels that are up there, they'll say, the response will be, well, there's no long-term studies of this, there's no long-term studies of that, right? So is it probably typically different groups, because it's probably not, I don't know about in the interest, but it's probably not financially motivational for the companies that are trying to get the drugs to market. They're probably moving on to other things rather than doing long-term studies.
Can you give me a more specific example? I'm not sure I understand.
Sorry. No, I mean, I guess there was nothing specific. It just, like I said, when it seemed like to hear it a lot when people would ask about, a particular drug and like the panel would respond, well, there's no long-term. I mean, it doesn't seem like there's a lot of long-term studies of anything, of how these drugs, like methotrexate, how, if you're taking it long-term, how does that affect like DM patients, or if you're taking, any other drugs like long-term, how does that impact?
Yeah, so there are different ways to answer that question. What are the long-term studies? Well, there probably isn't a long-term study because, how would you fund a 20-year study or 30-year study on methotrexate? And with newer drugs, there generally are no long-term studies because the drug has not been around long enough. But to answer the question about something like methotrexate, we don't have long-term studies, but doctors have long-term experience. It's just not in a study. Okay, it's stored up here. It's not in the typical clinician. They would say, well, based on my experience, based on what I've seen, this is what I know about this drug. Okay, so, but when you ask a question to a panel of experts on stage, they're going to take a very conservative approach to answering your question. They're going to try not to say anything wrong. Okay, so sometimes we don't always say exactly what's on our mind because we just want to make sure we don't say anything wrong. Okay. All right, so phase 1, as I said, it's usually a first in human, means this molecule has never been given to humans before, except through this study. And the main purpose is to establish safety in humans and to establish a dose. And usually we're trying to say, hey, we would like to give this dose, but we would like to give 10. But before we just run straight into 10, why don't we start at 1? If 1 looks okay, why don't we go to 2? And then maybe we'll go to 5, and then maybe we'll go to 10. And sometimes we'll say, let's just push it up a notch even further. Let's take it up to 15 or 20, okay? Just to see if we see anything. We think 10 should be good enough, but sometimes we'll play around with the dose just in case we need to go a little higher. So these are what we call dose-finding studies. They're usually small scale. In rare disease, it's 10 patients probably. It could be a phase 1 study. Then we move on to phase 2. That's a larger study. And usually a couple of doses have been determined to be our target. And we start monitoring more safety, larger number of patients. And we start to ask questions like, does it work? Do we think that we're getting an effect? Okay. Small numbers, though. And a lot of times you will get an effect, even if it's not a real effect, because there's a lot of times these are placebo effects. Okay. But when you see them, we start to take note of it, and we start to think, well, if we were to do our next experiment, what type of effect would we be looking for? Do we want to test the arms? Do we want to test the legs? Do we want to see how strong the legs are? Do you want to see how good they walk? And then phase 3 is usually the the final test. It's the final exam, okay? It's a big study, and big depends on what kind of disease you're talking about. If you're talking about diabetes, thousands of patients, right? Diabetes, heart disease, much more common, right? Talk about rare diseases like myositis, you're lucky if you get a couple hundred, okay? But it's large to try to be able to establish that you have statistics that can show you that this is not some random thing where we got lucky. I should say the drug got lucky. Nobody's getting lucky if a drug that doesn't work gets approved, okay? But if the drug sort of rolls the dice enough time, if you roll the dice enough times, then you could get, if you roll the dice 10 times, it's possible you get 6 every single time. It's possible. It's not likely, okay? So these are large studies to make sure that you've rolled the dice enough times to make sure it's not just a fluke. So repurposed drugs usually can skip to phase 3, or sometimes they'll start with a phase 2 study just to make sure if it's expensive, they don't spend too much money on a phase 3 study. They'll do a small phase 2 study, and if they show an effect, then they'll go to a phase 3 study. All right, just a few more slides. So rare diseases, it's difficult to recruit enough patients to get the statistical power to show that drugs work. And so even drugs that might work a little bit may not actually pass this test. So typically with rare diseases, we are looking at ranges of 100 to 400 patients. We want that statistical p-value, and so we say p needs to be less than 5%. Means that it's got to be something where it wouldn't have just been a fluke and happened. It would be only 1 in 20 chance that would happen just as a fluke. Okay, meaning that if you did the study again, maybe you would get the opposite effect. Okay. So, if that value is less, that means it's highly unlikely that this could have been just a random occurrence. What are the chances that you would get a positive finding in a drug that actually doesn't even work? Okay. That's what the p-value means. So with smaller numbers, you run into this problem, rare diseases, smaller numbers, you run into this problem where drugs that might work but only have a modest effect, okay, would probably not pass this test. So we're probably, missing out on some drugs that might have a little bit of effect. But the good news is that drugs that do pass this test generally are drugs that have a strong effect. All right, and then finally, why participate in clinical trials? Why should you, as a patient, participate in clinical trials? Well, I want to impress upon you that you participate in clinical trials because it's a selfless decision. Okay, you're doing it to help everybody, including possibly yourself. Okay, but you're helping the greater good of society and everyone else who has this disease. Okay, that's the way you should look at clinical trials. So it's an experiment, You're a guinea pig, so to speak, okay? If you go in it with that mindset, then you'll have a healthy attitude towards the clinical trial. If you go in there thinking, I want to get something out of this clinical trial, it's probably going to be sorely disappointing. Okay? So a clinical trial should not be viewed as a way of getting a treatment. Now, sometimes that's not totally true. Sometimes we'll say, we'll give you a placebo, but after a year, we'll put you on the treatment, assuming that we still think the drug works. Okay. So there's a 50-50 chance you might get placebo, but after 1 year, we'll put you on the drug regardless. Okay. A little bit of a reward for having been, a good sport about being in the clinical trial. So sometimes we do things like that. Okay. But you also have to realize there's a chance that the drug doesn't work at all. Okay. And at the end of that reward that you thought you were going to get, okay, may turn out to be something you don't want anymore, you know. So if you participate in a clinical trial, you should follow all the instructions, be very compliant, because this is an experiment, and the experiment will likely lead to data which will be beneficial for everybody who has this disease. And you want that experiment to be done well, okay? You don't want to negotiate and say, oh, you know, but I can't do that. I don't really, that's inconvenient. Then don't participate in the clinical trial, okay? Because you don't want to mess up the data, okay? Other people will participate in the clinical trial and get clean data. We don't want bad data. We want good data. Okay. So we do want patients to participate, with rare diseases to participate, because it's hard to find patients. You know, I can find a whole lot of diabetics to participate in a diabetic clinical trial, but it's hard to find myositis patients who are exactly what I'm looking for. So if you feel like you can, you know, that if you qualify and you feel like you can, you know, follow all the instructions and all the things that we want, then these are the types of patients we want for clinical trials. And that is my last slide, and I'm happy to take questions.
We have hesitated to go into clinical trials because the course of treatment that we're on seems to be working. Shall I repeat that?
Yes.
We've hesitated to go into clinical trials because the course of treatment that we're on seems to be working, and wouldn't we have to stop that course of treatment during the clinical trial?
Yeah, so that depends on the diagnosis and the treatment, but that's a perfectly valid reason not to participate in clinical trials. But, you know, sometimes there's a good reason why you might consider another course of treatment, another clinical trial, even though you're stable. That's unusual, but there have been situations where people will go off treatment because their treatment they think might lead to long-term toxicity. So there are a lot of different reasons, but I think what you're saying is very valid.
The age, people like us that's in their mid-70s, you don't want someone in the mid-70s who may not last another 10 years.
So that's not necessarily true. It just the person who's designing the clinical trial will determine those parameters, age, for instance. Sometimes we're looking for clinical trials to recruit patients who are older because those are the people who get those diseases. So I think it really depends on the disease, it depends on the drug, it depends on exactly what we're trying to capture.
Can you talk a little bit about orphan drug status? I sort of liken it, well, I understand
Just hold it up.
in the IBM or Withdrawal trial, orphan drug status kind of meant getting a fast pass at Disney. Went to the head of the line, didn't take quite as long, won't take quite as long, but that doesn't happen all the time. And I understand there's some incentives for the companies that may be sponsoring, but also it's a fast track.
Yeah. So orphan drug status typically is more financial rewards. So you can have exclusivity of the drug, and you know there are other designations like breakthrough and those types of things where you can actually meet with the company can meet with the FDA a little bit more quickly than the regular timeline. So there are a number of different statuses. I don't think that orphan drug status or breakthrough designation or any of these things mean that the process is any less rigorous. I think it takes things, takes out some of the wait times. So yeah, I think you're right. It's like getting a fast pass at Disneyland. You get to cut to the front of the line, but you know, you still have to last the whole ride, okay, without throwing up.
Do you know how many drugs are in the development process now, specifically for myositis, or how many trials in total there are? And how do we find out about them? I know they had the panel yesterday, but as they're coming up, how do we find out about them so that we can sign up?
So probably the best way to look, and I will tell you this because this is one of the ways I look, is to go to clinicaltrials.gov. Clinicaltrials.gov, and you can do a search based on your disease. This is true of any clinical trial, any disease. You know, it doesn't have to be myositis. You can do a search on, you know, myositis, IBM, dermatomyositis. You know, and sometimes you'll find that the disease you're talking about might be called something a little different than what you're calling it. So just make sure you know all the different names of your own disease. Okay, so that you know, because some companies may call it one thing and another company may call it another thing. But that's one way that you can capture that.
Oh, so I was just going to make a comment from the company side of doing clinical trials. So I work at a small biotech company. So we're running clinical trials in various diseases, including myositis. You know, ultimately, and as Dr. Shieh said, you know, they're all going to be listed on clinicaltrials.gov, but most companies, especially in the rare disease space, will make sure that they let all the advocacy groups in that area know that there's a clinical trial ongoing. So we'll be proactively trying to reach out to the community to let the community know that that's happening. That can also be through social media as well. I also just wanted to go back and make a comment about just the timing of clinical trials, and yeah, even though it is kind of it's a fast pass sort of at the review stage of the drug development process. You know, back to what Dr. Shieh was saying, it's really difficult to find patients to participate in clinical trials. So your enrollment timeline can be 2-3 years to get, you know, 50 patients into a clinical trial, and then you have to observe them with the treatment for another year. So it adds up. So the faster we can get people in and do that, the faster we can get drugs developed.
Thank you. Those are good comments.
How do you guys choose participants for the clinical trials?
How what?
How do you choose participants? Because I have gone to clinicaltrials.gov and emailed several people. No one ever asked, I just informed them who I was and what my disease was. And I was told I'd be put on a waiting list, but no one ever asked anything about anything else. So how do you choose participants? And the ones that I know who aren't part of them is because of the position. So how do some of us who don't go to Mayo Clinic, Cleveland Clinic, Ohio State get into clinical trials when no one's asking questions?
So I think your question was, there's a rowdy group over there. We're having a little too much fun. So yeah, I think your question was, how do we choose people for clinical trials? How do we select the patients, right? That's and you're describing some of the struggles you've gone through to try to be a possible candidate for clinical trials, and it's not been particularly successful, right?
Correct.
Okay. So I'll make a couple of comments about that. First of all, it could be really hard to participate in clinical trials because it's so popular and they don't have that many slots. So I think we ran into this issue for those of you who are in the IBM disease space. You know, the arimoclomol study, everyone's like, how do I get into that study? How do I get into that study? Okay. I see a few nods in the room. So, and you know, that's, like I said, I mean, we're trying to do our best. If we say we need only 50 patients, if we say we only need 100 patients, and then 500 people are calling us, I mean, it's, you know, there's just a limit, right? So sometimes what happens is that the doctors who are investigators will probably just be picking from the patients they already know that are in their clinic, okay, and pull them in, okay, because they already know they're going to qualify, as opposed to, and they have to look through this list of inclusion-exclusion criteria. Now that's a moving target because, you know, I've been working in clinical trials in other diseases like muscular dystrophy, for instance, and like about 10 years ago in muscular dystrophy, people were also fighting to try to get into these studies, okay? But then now, there are a lot of different clinical trials. There's a lot of companies that are involved in the research for that disease. And now those companies are begging patients to go into their clinical trials. So it's like the opposite problem. And I do feel like myositis is sort of, you know, one of these diseases which is now getting close to prime time, you know, in terms of it's getting the attention. So we might be seeing a lot more interest. Maybe the science is maturing. It's reached a point where we're going to be able to study patients with this disease with A, B, C, D, and E, and then you're like, I don't know which one I should go into, you know? Which one's better, A or B or C? I don't know, you know? And then you'll go and you ask your doctor, and you're like, the doctor's like, I don't know, you just pick one, you know? And you know, you qualify for all of these, we'll enroll you. So it really depends, you know? So I hear your frustration, because, you know, we, being the investigators sit on the other side, and of course we struggle because we get people who are calling us and asking us to, if we could screen them and put them in the clinical trial too. And you know, we get a lot of calls and we have to say no to a lot of people.
Could I offer some thoughts on that?
Mm-hmm.
I did get selected for the Withdrawal trial. There was a waiting list of over 100 patients for 10 slots at the facility. I was a pest for my doctors, and I called them, well, I saw them all the time. They know who I was. They know I showed up for appointments. They knew I would make the commitment. I still had to jump through every hoop. Dr. Damansky had to review my blood and my files. I had to do all the tests. I had to do the 6-minute walk. Can I stand up? Could I do this? You know, you don't just get to say, hey, I'll do the clinical trial. It's not that easy. It is a commitment. And you have to be the perfect guinea pig. Not everybody is. I just thought I'd have my 2 cents.
Yeah, those are good comments.
Yeah, you don't have the opportunity to even try it. You say you selected and it got to be special, but they got to call you first.
I called them all the time.
Right.
So did I.
But you know, on the other hand, I also want to point out that it's not necessarily treatment. You could be on placebo.
Yes.
Okay? And let's say the drug has a slam dunk. It works. Then we'll be able to give it to everybody. Okay, so that's, you know, this is, you know, it's the process is the process. Okay, the process is to get from point A to point B, and point B is hopefully that the drug works and we can prove it works and we can start giving it to everybody. So, you know, we as investigators are just trying to find patients who would be the perfect patients to prove the drug works if it works. Okay? And everybody will benefit from a drug that works, that gets approved.
What does location, physical location have to do?
Physical location.
Like you're in Los Angeles, and I live in Texas. So I mean.
Yeah, it's harder. Yeah, it's harder to participate in clinical trials if you have to travel a distance. But that's not a deal breaker. It just makes things more difficult. Yeah.
I'm interested in the phases that occur before a drug will get to you. What kind of feedback you as a clinical trial investigator can supply to the processes before you would receive a drug to test? I mean, is there any feedback you supply in reverse? Say, I'm looking for this, or help them narrow down so you can have some more control over what you receive, and therefore your general work is improved.
Yeah, I mean, generally speaking, companies that are developing new drugs, so once again, I'm talking about new drugs, they will ask experts in the field for their opinions about how they should do their clinical trial. What is the best scale? What's the best outcome? Should we do it for 1 year? Should we do it for 2 years? You know, who should we be recruiting? Patients who can walk 100 feet, patients who can walk 10 feet, patients who, like, how good do they need to be, you know, when we do the study, okay? Do they, you know, if it's dermatomyositis, you know, how bad does a rash need to be? What do we need to be looking at? Do Do we need to look at the rash in the back of the head or something like that? There are all sorts of things that you can be looking at. And the companies don't have the experts always in their company, and they will come and they'll ask people like us, medical advisory board. So lots of times when we give talks, you'll see we have all these disclosures that we've consulted for these companies, because they come and they ask us. And we don't give out our advice for free unless it's to you guys, you know? Right.
I noticed in your talking about outcomes out of phase 3, you were statistically based. But out of phase 2, you just said you needed a significant result. How do you determine it if you're not using statistics?
So you do use statistics in phase 2,
oh, you
but it doesn't necessarily have to pass the 5% test. Okay. But if it looks like it's going the right direction, you know, and maybe if you did the statistics, you said it was a 10% chance that this could be a random occurrence, you could say, well, but it looks pretty real. A company might just trash the drug at that point and just say, we're not moving forward. Okay. Or they might say, you know what, we really do believe in this drug. We're going to move forward with a phase 3, and we're going to make sure we recruit enough patients so that 10%, if it's a real effect that's going to happen again, it's going to drop down to 5% or less.
Just real quickly, I understand about the inclusion-exclusion criteria, but how is it that a lot of the studies arrive at a 10 or 20-person clinical trial? Especially given, because even for this conference, you know we have probably 500 people that are affected with some variation of the disease. So why would we limit it to such a small number given that population size?
So it really depends. I mean, if patients have other diseases, that would usually exclude them. Okay, it depends on how stringent they put in those things. For progressive diseases like IBM, for instance, we probably want to recruit patients who still have a significant amount of muscle, okay, because we're probably talking about preservation of muscle. But that may not be true with patients with one of the other myositis, like necrotizing myopathy, for instance. And these patients, maybe you want to recruit the patients who are really weak, and you want to show how much stronger they get, you know? So we may be looking for a specific group of patients, you know? One more question? We got one more question, 2 minutes.
Does the patent process or big pharma affect your clinical trials?
I don't think that it doesn't, what do you mean does it affect it?
Well, are you limited in what you can do in your trials by the bigger pharmacy companies being ahead of you and not wanting you to do things?
So I think, so it doesn't affect me because, you know, I am just recruiting patients for a clinical trial that may be company A. A lot of times these companies, A, B, C, D, different companies, they might be competing companies, but they'll all come to me because I have the patients, okay? So in some ways, I may have, you know, some type of consulting relationship with them, but oftentimes it doesn't affect the investigator because what we're doing is we're just trying to bring drugs to our patients, and we'll work with any company that has that goal. So, does that answer your question? Okay, well, I thank you for your attention.